Alright guys, everybody do me a favor, get out your Weinberg data from the paper, and we're going to do the class data. It looks like some of you guys wrote it down and already calculated it. Wow, I'm impressed. Good job. That I hadn't posted the textbook pages for today's reading quiz. I was just going around thinking, oh, I just need to post the workbook pages. So that's why you don't have a reading quiz today, because that's all on me. But next time, tell me. Be like, hey, miss, by the way, you're slacking during lunch. Can you do that for us? So just let me know next time for sure. I will definitely not take offense to it, unless you say it really rude, then I'll be hurt. But if you ever see our calendar, what have you noticed about this unit? We're crazy changing it up, and that's not like me at all. You can ask my students last year, but that's okay. So this is what I have right now, right? I'm gonna change it. We go really fast, and so if I need to slow down, I'll slow down. So I never put tests on Fridays. Let me repeat that. You never put tests on Fridays, okay? So I had the test scheduled for Thursday, so now the test is going to be the following Monday. For now, I could change my mind and say, you know what, these kiddos need more time. I could change it to the following Tuesday. Just depends on how we're going. This is a monster unit. So you've got 4 main themes in your class on your exam. Evolution is 1/4 of your information on your AP test. So it's huge, really, really big. So this is what I have. You guys are going to have that reading quiz on Monday. So during lunch today, I will post textbook and workbook pages for you, and you will have that reading quiz. I'll show you on your printed notes what it covers. And then right now, I have your lab due. It was formerly due on Tuesday. I'm going to give you guys a whole week to get it done. So as long as you have your lab due to me on next Thursday, okay? And then I think I'm going to save the multiple choice quiz for the same day. So make sure that you don't wait until Wednesday night to do both, right? Because that's kind of heavy, but I think I'm giving you plenty of time so that you don't have to. And you also don't have a graph on this one, and we're gonna hopefully knock out that data together right now. And then let's review Friday and plan to test a week from Monday, okay? All right, guys, so let's look at this data. I put it on Google Classroom for you last night. I thought y'all did a great job answering my questions, showing me that you knew how to calculate Weinberg. So we've got quite a few absences. Let's just talk about this really quickly. So here's the Weinberg fish data. That's right, good. So notice my little disclaimer. Make sure you use this data to complete the entire chart on your lab assignment. Believe it or not, last year I had some students just write this down for class data and think that they were done. But that's not the case. Alright? So we're going to write that down on this piece of paper, and then we're going to talk about how our calculations should look. So if you don't have that, if you didn't jot it down right now, no big deal. Let's look at it together up here. And we're going to use our calculators. If you're somebody who has already done this calculation, you're just that awesome, just follow along to make sure you don't have any mistakes. All right, so that gold, I just want to reiterate, if you have a goldfish, okay, what's their genotype? Tell me, louder. What kind of S? Homozygous what? Recessive. Okay, so I just want to reiterate that. Now, if you pull a brown pretzel fish, do we know that organism's genotype? No. Perfect. So we know they are either homodominant or heterozygous. Okay? And that's just simple Mendelian traits, right? The other thing that I want to bring up is Q squared represents a genotype or an allele? A genotype. Which genotype? Two little f's, homo recessive. Okay? Q represents what? What allele? Little f. P represents big F. P squared is homodominant. And 2PQ is heterozygous. Okay. So I did that for 8th period yesterday because I felt like they were a little confused. So I just wanted to reiterate that with you guys, make sure we're on the same page. So if you don't already, copy down this data from Google Classroom with me. 109, 80, 57, 45, 44. Okay, our next one, 111, 140, 163, 175, 176. Okay, so that's what your data should be. Now, when we did class data or group data yesterday, it was really simple because we had 10 organisms in the population. So whatever our gold was, it was just put a decimal in front of it, right? Like if you had 6 gold, what was the frequency? 0.6. If you had 9 gold, then the frequency was 0.9. Is that the case with our class data? No. Okay. So Leilani, how do I go about finding Q squared? Can you tell me? How do I go about finding it?
Well, for the other ones, for chart 1, it was like 4 and 6, so I put 0.4.
Right. But now, we don't have a value that's divisible by 100, so how are we going to do this? And if you don't know, that's okay. We're going to work on it together, okay? Who feels like they may know? Michelle? What's our first step? Like, what's the first thing I need to do? I need to add these together to get my total what? Population, right? We've got a population, add together 109 plus 111. If I add those together, the total population of fish, who's right here with me, is 220, okay? So we added those together. So if I need to help you with that, we'll put total population is 220. Does the population size remain the same throughout each generation? Yeah, absolutely, okay? So now I've got my total population size. Kent, what's next? What's my next step? Perfect. And we're going to change that percentage to a frequency. So how do I do that, Kent? What do I put in my calculator? 109 divided by 220. Absolutely. So I'm going to put that in this little box just to help you guys figure out the rest. 109 divided by 220. How would I round that, y'all? 0.5. And I just want to reiterate, if you get a value that's like 0.99 for P plus Q or 1.01, that's acceptable. We're not going to worry about fixing or manipulating rounding for literally 0.01. Okay, Aaron, not Avila, but Arizola. How do I do the next one? 80. Perfect. 80 divided by 220. How do I round that, you guys? 0.36. Okay, can you do the rest? I think so, right? I think so. So if you look at, without even looking at or calculating the frequency of Q, what is happening to the frequency of the F allele over these generations? It's decreasing significantly. So that tells us that what is happening? Evolution. Evolution is happening. Do we know which of the 5 mechanisms is causing it? In the lab we do. Anani? Yeah. Yeah, because you guys were selecting against the gold phenotype. So we do. In an actual simulation, in an actual ecosystem where we're taking a population, would we know for certain what mechanism is driving it? No, we would have to do a little bit of investigation and think about, okay, is there some gene flow going on? Maybe the breeding population isn't large enough, but we know, in fact, which one of the 5 hasn't been met for this one, okay? So for the lab, remember it's due next Thursday. Finish all of this. There's no graph. You just need to put that data on there. Those data tables, make sure they're nice and large, hopefully at least a half a page for me. If you ever need to turn your lab notebook, for instance, because there are quite a few columns, if you ever need to turn your lab book, instead of doing it like a portrait, you want to do it landscape, that's totally up to you. That's acceptable. Just make sure you keep it on the right side of your journal. Okay, perfect, guys. Put that class data away for me. I want to talk to you about how Hardy-Weinberg is going to be used on your exam. We have not finished with Hardy-Weinberg. We're going to visit it every single day for this unit. I loved this slide yesterday. Wow, I don't even have a mouse. Here it comes. Okay, I loved this slide yesterday, so I'm going to call on you guys and you're going to walk me through this. Because y'all did a really good job telling me that you knew what was going on with this. So, Alicia, 99% of the time, which of these 5 variables are they going to give you in the problem, honey? Q squared, right? Perfect. So we know that they're going to tell me Q squared first. Carol, how come I can easily find Q squared compared to the other 4?
This is the one that is noticeable.
It's the one that's noticeable by what alone? By the phenotype, right? Because I don't have to do any genetic testing. I don't got to run any DNA tests. I can just look at that phenotype and know this little white cat, he's really cute, right? He is going to be homozygous recessive. Okay, Maggie, I've got Q squared. What do I do next? Which of these other 4 am I solving for next? Q. How do I find Q? Square rooted. Good. Kasia, what's next? I got Q. What's next? How do I find P? Subtract it from 1. Perfect. Okay. Eve, I've got P. What's next? Find P squared, right? I'm going to square P, and I get there. And then Tosin, the last one? And then I multiply 2 times P times Q, and I've got everything I need. Could you indeed check your work? Yeah. If you see something that equals P squared plus 2PQ plus Q squared equals a 1.01, is that okay? Yeah. What if it equals 1.2? You made a mistake, right? If it equals 0.8, you made a mistake. And I saw some people make mistakes. You know what they did yesterday? They did 2 times P times Q squared instead of doing 2 times P times Q. And remember, the order of those columns is in fact for a reason, to help you go in that direction. Okay, so let's do a practice problem. I'm going to call on different people this time. This one's nice and easy, right? Annalisa, what's the population size? So that's going to be easy. If I've got 84 black and 16 white, honey, how many, or I'm sorry, what's the frequency that we can solve for right away? The white, what's the value? Is it PQ, P squared, 2PQ, or Q squared? The Q squared, perfect. What is it? Without even needing a calculator, what is it? 60. But in frequency, it's going to be 0.16. Love it. Okay, we got it. So we know the Q squared right away. It's 0.16. Do we need a calculator for that? No. It's nice and easy. Okay. Now what are we going to do? I want to hear from Madison. What's next? Solve for Q by square rooting it. Perfect. We're going to get 0.4. Okay. Let's do the next one. Miss Ariel, what's next? Do you need a calculator? What's the answer? 0.6, right? She's got it. All right. So now we need genotype frequencies. So without asking you guys, you would just go in, assume it's in Hardy-Weinberg, you would plug and chug, and you would get these values. Okay, so I think you guys know how to solve things in a very simplistic way. That's the first level. Now you've got to analyze. Okay? Miles, what do these frequencies tell me about the population?
Most of the population is heterozygous.
Yeah, did you guys hear him? That was perfect. most of the population is what, you guys? Heterozygous. Okay, Michael, what's the lowest quantity in the population?
The white.
Or what genotype?
Homozygous percent.
Nicely done, homozygous recessive. Y'all are doing good. Let's take it another step. So this was our population of cats. Now we're going to look at a second generation. Here's our second generation. Whoa, right there. Okay. Ah, no, stop. Okay, so if we go from the top to the bottom, you can talk about a lot of stuff. Look at it for a second. This is not as easy as you may think. Okay, I need you guys to think about what occurred from generation 1 to generation 2. There's a lot of things that could be said, things you might not have already thought of. Okay, Sophia, what can you tell me? What changed in generation 2? Yeah, so what does that tell me about the fitness of homozygous dominant? It's greater. We call that an advantage, okay? It has some type of advantage. Do we know what that is? No, but we can make some assumptions about what it could be. Okay, Aaron Avila, tell me about my white cats.
It remains the same.
They remain the same. So Tosin, what does that tell me about the mating preferences of white cats?
They just mate with themselves.
Yeah, well, yeah, with other white cats. I knew what you meant, Hank. I knew what you meant. Yeah, the white cats only like white cats, right? Perfect. Excellent, excellent, excellent. Okay, now look at this set of data. That was one set. Here's a second set. Let's assume that was original, and here's second population. Okay, Genevieve, tell me something that you see. What happened, honey? The heterozygous, right? Okay, they went up. They went from, in an original population, they were 0.48. Now they're at 0.64. Okay, you ready for this? This is really common. This is called a heterozygous advantage. They have a heterozygous advantage. So as a carrier of that recessive allele, they've got some type of fitness advantage. You guys ever heard of sickle cell anemia? Okay, we're going to talk about sickle cell anemia next week and how those individuals who are not homodominant, who are not homozygous recessive, heterozygous recessive gives you sickle cell, but if you are heterozygous, you're got an advantage. You might have 50% of your red blood cells being sickle shaped, but you have an innate ability to be less likely to get malaria. That's the advantage. So I'll reiterate that. All right, malaria is a disease that's carried by what? Mosquitoes. And where are the majority of those mosquitoes found? What continent? Africa, right? Okay, so do you guys see how calculating it is the first layer? And then you've got the second layer, which is doing what? Analyzing the trends, right? White cats only mate with white cats, that heterozygous advantage. So we'll get into that. Everybody do this. Let's do, we did Hardy Weinberg. Let's do, I'm gonna do this one instead. Sorry, y'all. Perfect. No, I don't want the review one. I want the quiz review one. Aha, found it. Okay, we need to get, I'm skipping all of that because I don't like those. Bam, right here. Write those three down. You probably already did last time, one time. So we're back on our chapter 21 notes. 3 major factors that alter. When we started yesterday's Hardy Weinberg, we talked about 5, right? We talked about 5 different mechanisms. We talked about these 3. The other ones we talked about were non-random mating and mutations. Do y'all remember that list you made? So while mutations and non-random mating have a significant impact on changing the allele frequencies, these are your main 3. Okay? These are your 3 what I call heavy hitters. These are your 3 heavy hitters. So, good thing is we already know natural selection because we covered it last weekend. Let's go with Erica. What's the keyword for genetic drift? By chance, I like it. Renee, what's the keyword for gene flow? Migration. Y'all are hitting it out of the park today. Awesome. So let's keep going, you guys. Natural selection, differential reproductive success. Okay, Tosin, what's placing the pressure pressure on those that are deemed fit?
What is it?
Natural selection, what's placing the pressure on those organisms that are more fit? The environment, perfect, okay. Those that are more fit are gonna pass on their traits more frequency, and we should see those alleles increase in greater proportions. Now let's get into genetic drift. I promise I'll put all this on classroom, you'll be fine, okay? We talked about it being by chance, random events. Give me examples of by chance random events. Yeah, Ani. A group of bugs being stepped on, a good one. Yeah, just tell me. Yeah. Natural disasters. Perfect, natural disasters. So if you need to write some volcanoes, hurricanes, typhoons, any of that, then you can go ahead and put that down. All right. Now you're gonna learn today that there's two types. Two types. Well, no, I think it's two types of gene flow, right? Yeah, two types of gene flow, but genetic drift by chance. Here's a really important thing to think about. Genetic drift tends to reduce genetic variation in small populations. So write down, small populations are more susceptible to negative impacts of genetic drift. I'm gonna re-say that, okay? Small populations are more susceptible to negative impacts of genetic drift. Because think about it, if you've got a small population of only 100 crickets, and a volcano wipes through and eliminates 90 of them, is there a chance that there might not be a lot of genetic variation remaining? Considerably, right? They could have only left all the homozygous recessives. And if they only left the homozygous recessives, are we ever going to have that dominant allele come back? Only way would be with gene flow or a mutation, right? So, small populations are really susceptible to negative impacts by genetic drift. That's really important. All right, so here's a summary. Significant in small populations, you just talked about that, can cause allele frequencies to change at random, lead to a loss of genetic information, and can cause harmful alleles to become fixed, right? I kind of gave you that example with the crickets. If the homozygous recessives are all that remain, if they're harmful, what's going to happen to that population? Tell me, guys. They're going to die, right? They're not going to make it. All right? So, what do we know that how's natural selection different? I talked to you about this last unit. How's natural selection different? Genevieve, how's it different? Or you know what? I picked up Genevieve. Daniel, this one's yours. How's natural selection different?
Because it's like preserving like the fitness of the
Okay, but how does it impact the population always? Natural selection. Natural selection always does what to the population? Increases the fitness, decreases the fitness, both? What are you thinking? Jackie, help him out. Always. Remember, you guys, natural selection always causes adaptive evolution. Does this look pretty grim to you, this summary of genetic drift? Look at how negative this is. Significant in small populations, change in allele frequencies lead to a loss of genetic variation, harmful. Does it look so grim when we think about natural selection? No, natural selection is amazing at helping improve the fitness of a population. How frequently, Daniel? Always. Always. Always, 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 always. Okay? All right, guys. Let's talk about the founder effect. Okay? And I guess I have this right. Yeah. Okay, there's two types of genetic drift. I was second guessing myself. I was like, I think I'm saying that wrong. Two types. Write down founder effect, keyword isolation. Founder effect, isolation. So this happens significantly when you are looking at islands, okay, from the mainland. Isolation. This also happens with human interference. Okay, we'll talk about that as well. So founder effect is when a few individuals become isolated from a larger population. Sometimes that can be a man-made structure. All right, over time it can be a geographic range that's going to isolate one population from another. The Great Wall of China is an example, right? When they constructed the Great Wall of China, we had two species that were interbreeding. Then they separated, and we had speciation occur with different trees. But allele frequencies in the small founder population can be different from those in the large parent population. So that's when we have one population become isolated, and then we have a change in the allele frequencies. It could be good or bad, but it's usually bad on the small populations, okay? The second type of genetic drift is called the bottleneck effect. The bottleneck effect. All right, keyword for bottleneck, I want you to write down near extinction. Near extinction. So this happens commonly if we find a population is being poached, right? Individuals are killing animals for their resources, right? We saw this with elephants and their ivory tusks. We've seen this with certain types of large cats because we want their very soft and luxurious fur. So this happens quite a bit. If we find that a population is going near extinction, what do we as humanitarians tend to do? Save them. Yeah, we try to save them, right? So we try to take that small population in an isolated habitat, try to get them to breed and get them back up to where they need to be, all right? So if you've got bottleneck effect, it's a near extinction. What's the population size? Small. So are good things or bad things probably going to happen to this population? Bad. Bad things are usually going to happen. So here's the example that they give you. You can see in this original population, how many different colored beads do you count? Three. I count blue, white, and yellow for a total of 3. Bottleneck effect, what's our keyword? Near extinction. So we have this huge population. Now it's dwindled to 6 individuals remaining. 6 individuals left. Now how many phenotypes do we have? 2. Okay, it could have been that those yellows were good, right? It could be that these overwhelming amount of remaining blues are negative. Either way, they're more susceptible to negative impacts due to the change in allele frequencies. So the Florida panther is one of those examples that they talk about that went to a near extinction because of their habitat destruction that we intended as humanitarians to try to bring back to a greater population size. It takes many, many generations though in order to get them there, because what we were finding is that the panthers that remained, they were infertile. A lot of them were infertile, so we had to keep trying and trying until we got one that was actually fertile. I think they got down to as less than 10 of those Florida panthers, if I recall correctly. Anybody read that in the book? Well, you couldn't have, because I haven't posted it. No, I think maybe I did post this one. I'll figure it out. Okay, let's do gene flow. Gene flow. Movement migration. Movement migration. Okay? So it tends to do what to genetic variation? The bolded terms in this PowerPoint are most significant. Tends to reduce. So things aren't looking too good, right? We looked at this summary of gene flow. Was that good or bad? That's bad, right? That was pretty bad. It's our genetic drift. Gene flow is not looking much better, right? So when we have a movement of alleles, immigration, right, with an I or an E, we're seeing that it actually decreases or reduces genetic variation. And that's a negative impact. So let's take a look. It can decrease the fitness of a population. All right, consider, for example, the great tit on the Dutch island of, I don't know how to say that word. Anybody know where this is, this Dutch island? Anybody know? Leland? Golly, I'm such a South Texan Spanish speaker and English speaker only. Okay, so immigration of birds, let's talk about it. Birds are common with gene flow because they constantly migrate from the mainland, introduce alleles that decrease the fitness on the island population. So we've got some mainland birds, they decided to go ahead and travel to this Dutch island. Natural selection then reduces the frequency of these alleles where immigration from the mainland is low. All right, let's understand that a little bit better. Reduces the frequency of these alleles where immigration from the mainland is low, okay? In the central population, high immigration from the mainland overwhelms the effects of selection. Okay, so what that's telling you is the mainland gets an overwhelming negative impact from the immigration back to the mainland, and then the positive natural selection effects were overwhelmed, and now you've got a decrease in genetic variation. Because of what? Gene flow. Gene flow, okay? So gene flow and genetic drift, usually bad. Could they be good? Yeah, the probability of that, a lot less likely. All right, here's an example where a gene flow actually increased the population, the spread of resistance, or the spread of alleles for resistance of insecticides. So insecticides have been used to target mosquitoes that carry our West Nile virus, along with other detrimental diseases. Alleles have evolved in some populations that confer insecticide resistance to these mosquitoes. The flow of insecticide-resistant alleles into a population, so if you've got some resistant mosquitoes flow into your area, is that a good thing? Yeah, so here's an example of where it actually did have a positive influence on the genetic frequency of this population of mosquitoes. Not good for us though, right? We don't want those West Nile mosquitoes around us, but good for that population. So here again, I'm harping on Daniel, right? I'm sorry, Daniel. Natural selection is the only mechanism that consistently and always causes adaptive evolution, all right? We've seen all this. Yeah, we're good with this. Let's keep going. Here we go. This is on your quiz on Monday, so make sure you're paying attention. This is a little preview. A little preview here. So we've got two types of selection. Write down all three of them if you haven't already. You've got directional, destructive, and stabilizing. So you guys probably thought, oh man, there's only, natural selection, that's so easy, there's one. Now we've got three. We've got three different types, okay? Oh, I went too fast. Actually, I kind of want to do it with the pictures. Watch. These are your three, to write down here. Directional, destructive, stabilizing. It's going to be better to look at it with a graph than it would be written in words. I can tell you, you've got to know these graphs on Monday. Man, I'm being so nice. I don't know what's going on. I don't know what's going on with me. You like it? I kind of like it too. Okay, guys. What is our population according to these images? A population of what? Mice. Okay. Are these simple Mendelian mice, or are these polygenic mice that have a bunch of different coat colors? Like us, right? There's not just two skin colors in us. We have polygenic inheritance, meaning that from our 46 chromosomes, we've got to look at least 6 different chromosomes and certain gene loci in order to determine our skin color. Same thing with these mice. These mice are not simple brown or black, white or gray, okay? So we've got a range here. Notice the frequency of the colors. In the original population, the intermediate color, right, this kind of soft tan color, has the highest frequency. So that solid line is the original population. Now, the dashed line is our evolved population. So what you can do here, if you want to sketch these, which direction is having increased fitness? The direction to the right. So it's headed in one direction. The bell curve in this scenario has shifted in one direction. So, right? Miss Margot, which color is better now? It's not that tan, it's a now what shade? Darker shade, yeah. In this direction. What happened to the white little mouse, Renee? Yeah, natural selection got rid of him. In this population. Okay, everybody feel good about directional? All right, let's talk about destructive. That sounds negative, right? You're so destructive. Jose, what's going on in destructive? This is pretty significant here. What happened to my beautiful tan intermediate mouse? Yeah, he was selected against, right? So what are the two best phenotypes? The lightest and the darkest. So it disrupted, right? It disrupted the intermediate. So it selected, see the arrow? The arrow's telling you fitness
natural selection worked against bees. And look at the evolved population. We're not seeing any intermediates. Okay? On this graph, the white arrow pointing down, on the directional, we eliminated all of those light-colored mice. Natural selection worked against those light. And then our last one, guys, is stabilizing. Stabilizing. Tosin, talk me through this one. What's going on in this one? How does the evolved population, that dashed line, look different? Well, all you need to do, and you're probably not the only one. If you look at these 5 mice, right? You've got 5 total phenotypes. What is the only phenotype that's being selected for? The middle one. Yeah, we call that the intermediate one, right? And notice the arrows. They got rid of the super light ones, and they got rid of the darkest ones. So it's that intermediate one. What's happening to the frequency of the intermediate? It's stabilized. It is much more frequent, right? You can see that jump from the solid line to the dashed line there. Okay, so make sure you know these, okay? Natural selection can occur in any one of these ways. It just depends on the environment. Ideally, this is our population. Who can tell me why ideally this is our population? What is better about this population compared to those 3? Go say it, tell me.
Variation.
Excellent. More what, guys?
Variation.
More variation, right? So, hopefully this is what's going on because we've got more genetic variation, but we know we can trust natural selection. How come, Daniel?
It's always happening.
He's got this, you guys. He's got this.
Does it always increases the fitness?
It always increases the fitness. So if we have directional, destructive, or stabilizing, can we trust that it's good? Yes, we can. We can. It's good for that population. Even though there's less genetic variation, is that okay? Yeah, it's okay. It's A-okay. All right, so directional, let me read you the definition. Favors individuals at one end of the phenotypic range. So directional is like sliding left to right. Okay, destructive favors what ends? Both ends, and goes against the intermediate. That's where you have those 2 humps, like that. Oh my God, I just thought of Fergie, and that is awful. Anybody? My daughter heard my hump song, and she was like, Mom, I don't like this. I'm like, thank God. Oh my God, it's such a bad song. But when I was in college, boy, that was always playing. Okay, stabilizing. Think back to stabilizing. It favors the what? Intermediates. So you saw that bell curve get more narrow in favor of the intermediates. I think y'all did a really good job with that. This was so hard for my students last year. Okay? Ken's like, what? It's probably because they're just sucked at teaching it. It's probably my fault. Don't worry about it. Okay, we ready to keep going? We're almost done. Oh, did I go too fast? Come on, Michelle, you got a front row seat. I just threw you off with my humps. Should I play it to wake you guys up?
I drive these fellas crazy. I do it on the daily.
No? I don't know every song. I don't know every word. I promise.
They treat me really nicely.
Okay, it's Friday. Can you tell? It's not even 8th period yet, and I'm acting this crazy. All right. Genetic drift and gene flow do not consistently lead to adaptive evolution. Did we establish that?
Yeah.
Okay. I'm gonna call and pick on somebody again. I don't know why I'm doing this so much today. I wanted to hear from Anna E. Anna E, give me the example of gene flow that was good for the population.
The mosquitoes.
Nice.
The
Yes. The insecticide-resistant mosquitoes. Not good for humans, but good for that population. They were able to travel into a mainland that did not have resistance, and then they were able to reestablish some mosquitoes that had resistance, insecticide resistance. Perfect. Okay, let's do another one. Erica, give me the key word for founder effect. Huh? Isolation. Did you even look at your notes? I am so impressed right now. Nice, Miles. What is the key word for bottleneck effect? Not very good.
Near extinction. Okay, Genevieve, genetic drift, usually good or usually bad? Absolutely, good job, you got it. What is it, guys? Usually what? Usually bad, okay? Gene flow, Madison, what's our keyword? Migration, good. Leilani, give me, in your words, an example of directional selection. Explain it to me. I just taught it to you. Can you explain it to me?
Just where you go like left and right.
So it favors one end of the phenotype. Good, good. Sophia, give me an example of stabilizing. How would you describe stabilizing to a friend? Right, the intermediate species is favored. Good. Tosin. Destructive. How would you describe destructive selection?
Favors the ones with poor kinds of graphicals.
Perfect. Members at either end of the phenotypic range are selected for. What's selected against in destructive, you guys? Intermediate, because it dips down in the middle. Okay? Good job. Okay, let's talk sexual selection. This gets a little awkward. We're talking about mating success. My freshmen make it really awkward when I start talking to them about like finding mates and like, you know, yeah, see, Ariel's like, Miss, I got this. I'm mature. All right, you got two different types. So we're gonna get into this. This will help you a little bit. We're almost done, guys. But sexual selection is natural success or natural selection for mating success. So remember, one of the 5 conditions of Hardy-Weinberg is random mating. Okay, random mating is what we need. But most of the time in nature, are individuals selected for mates in a very like meticulous and picky way? Yeah. Have you met a woman? We're picky, right? We're really picky. And that occurs not just in us, but in many, many different species. Okay? So we've got intra and inter. Let's learn the difference right now. So intra is competition among individuals of one sex for mates of the opposite. This is guys dueling over a lady. Okay? So intrasexual, competition among individuals of one sex. It could be 2 women, right? But it's oftentimes in nature among other animals. It's often males competing for the opposite sex. A lot of those intrasexual encounters, do they often lead to death? Not really. It might seem like it, right? It might seem like it. At Marshall it doesn't. Ariel, I promise it doesn't. But oftentimes it is really just an act of, look at me. Look how strong I am. You want these traits. You want these genes in your children, right? Now, intersexual selection is called mate choice. When individuals of one sex, aka usually females, all right, I made that statement intentionally, we're more choosy overall in selecting our mates. That's not with 100% certainty, okay? Everyone's different. But the women, why do we get to be more choosy? And it's not because of, it's a little social, but it's also physiological. Think about it for a second. If a female animal gets pregnant, do they have a huge burden on them? Yeah, because in 90% of the cases, who's going to rear and care for the offspring? We are, right? Men, males, not just, don't think humans, think about every animal. Do they have a huge investment in offspring rearing? No, they don't. So they are not as choosy. They are naturally and psychologically and physiologically predetermined. I'm giving you really valid reasons, okay? I'm kind of a feminist, but they have plenty of reasons. It's in their evolutionary thought and inheritance to, be careful with my words, to rear as many offspring as possible because it increases their fitness. They don't have an investment, right? And most of the time they don't. They don't have an investment in the offspring. They simply need to reproduce as often as they can to increase the fitness of themselves. Because the more offspring they have, the better their fitness, the more their alleles are transferred. With Females, we are way more choosy. We've got a 9-month investment and we've got potentially 18 years or longer that we've got to rear this child. So we're very picky because we want to make sure that that kid that comes out is decent and not always in the principal's office. Okay, yay, we finished! And that's called the good genes hypothesis. We're good. All right, guys, y'all did so good today.